Okay, so we looked at blood types. Now let's figure out how do you determine blood type? Okay. So I know in class this was a little bit difficult to understand, but we want to think about you have a sample of blood. You have your patient's sample of blood here. Okay. This is your patient, patient one. I don't know. You can just call them, yeah, patient one. We need to know what their blood type is. So that's what you've been asked to do. Blood type, question mark. The first thing you want to think about is, well, what if I'm asking what their blood type is, what does that mean? What do I need to look for to help me figure out what that blood type is? Like I paused because I'm hoping you're like responding with, oh, I need to know the antigen. Okay, right? Oh, I need to know the antigen because that's my blood type. So I want you to think about if you need to know the antigen, what can help you figure out what antigen is present in blood? I'm just gonna give you a minute to think about that. Okay, I know you can pause the video, but I bet you don't. So we need to know what the antigen is. If we go back here and we see, oh, that's right, our uh our free-floating proteins. Oh, I can't erase this. Our free-floating proteins are our antibodies, and when they attach, that tells me I have an antigen present. Oh, maybe I can use that to help me, okay? This is not happening in the body, okay? So what's going to happen is you're going to take your patient's blood out of their body. Please take it out of their body. Okay. You take a little sample out of the body and you're going to use what's called a well plate here. And basically, you have a little depression in this well plate that's labeled A, B, and RH. Why? Ah, because there's an A protein or an A antigen, a B antigen, and an RH antigen that we have to check. If you're wondering why there's no O, remember O doesn't have an antigen. Okay, so you can't check for that one. You can check for it, but you can't check, you can't you can't put a well in here. Okay. So what I'm gonna do is I'm gonna take like 1 to 2 drops of blood, mostly 1 drop of blood, and I put it in my little well. Okay. And then you can buy, oh, I did this kind of bad. I'm gonna change my red color here. I should have realized that. Sorry. Um, you can buy something called anti-serum. Okay, it's like anti-A anti-serum or anti-A serum. So you have like a little vial of anti-A serum. So this is talking about class. The anti-A serum equals antibodies or A or anti-A antibodies. Why is it called anti? Because anti means against. Honestly, they could have just called it anti-A body, but whatever. So you can buy that little serum, okay? And then you can buy a little serum that is anti-B serum. Guess what it has in it? Anti-B antibodies. Then you can buy in one for RH. Forgot the I there. Anti-RH. These are called anti-D, but I just like to use RH. So the anti-RH serum has, guess what? Anti-RH antibodies. And I know what you're thinking, but you said the body doesn't have any RH antibodies. These are simulated. We're making simulated antibodies. Okay, so similar to what your body would create in terms of an RH antibody. What we're gonna do is in the little well over here, we're gonna put the A antibodies. And if it helps you, maybe don't do it like that. Let's just say, okay, we're going to put these antibodies in here for A. In the B well, we're going to put the B ones. And in the RH one, we're going to put the RH ones. Okay? And we're going to look for a reaction. There are two possible reactions that can occur. I want you to think about that. If the antigen is present, what reaction is going to happen? Let me erase that. I don't need that big giant line. If the antigen is present, you're going to have agglutination. Okay, so in this, it's going to get thicker and it kind of clumps together because the red blood cells will clump or cluster. So when you have clumping, that's agglutination. This equals antigen. I'm going to use my abbreviation here. I shouldn't do that. This equals antigen present. Okay. When you have no clumping, this equals no antigen present, so antigen absent. That's what's happening. So let me draw out exactly what's happening between these two things. Let's just give ourselves an example. Okay. We have a red blood cell here. Ooh. We're going to have a red blood cell here. And let's just say our red blood cell, right, has the A antigen on it. So when I put my A antibodies in here, you see how those A antibodies are going to attach and cause clumping. You just have to imagine a whole bunch of other red blood cells. Let's just say, though, I have another red blood cell over here. I have nothing on its surface. And I'm going to put this antibody, this A antibody, in here. You see how there's nothing for this to attach to, so that means no clumping. So that tells me that that antigen for A, this is my A antibody, right? My A antibody. It's telling me that at least for now, my A antibody is telling me my A antigen is not present. So then I can say, okay, well, my blood type doesn't have A in it. Or in reaction one, my blood type does have A in it. So let's do an example. Your example is patient one. Okay, so in the A well, I'm going to have clumping. Why? Because I already did it up there. Let's say in the B well, I have no clumping. And then let's say in the RH, I do have clumping. Okay, what is the blood type? I'm almost willing to not give you guys the answer to this to see if you can figure it out. All you got to do is go through and say, well, clumping means my antigen is present, so A. No clumping means my antigen is absent, which means, okay, I have A, but no B. And then for RH, clumping means it's present. Remember, when RH is present, right? When RH is present, I have a positive sign. So that gives me a positive blood. So think about what would happen if you have clumping in A, B, and RH. What happens if you have no clumping in A, in B, or RH? Those are the things you want to think about. This is how you figure out what the blood type is. This is something that you will do in lab. If you're watching this in the future, I hope you're still doing that in lab, okay? So let's look at compatibility next. How do you figure out blood type compatibility for transfusions?